All right, hello everybody. All right, hello everybody. My name is Andy Garza, and today I'm going to be talking and illustrating to you guys about Chapter 3, which consists of antigen capture and presentation to lymphocytes. So we're going to get right into it. So essentially, antigens and immunogens. An antigen is basically any molecule that can be recognized by the immune system. This antigen can then trigger an immune response, right? So it's an antigen. Let me change the font. Antigen and a person, which has immune system. Immune response, right? Okay, so it essentially can trigger an immune response, and immunogens are also a subset of antigens that can specifically induce an immune response, right? So these antigens have little things on them, and these right here are immunogens, right? They attach to the healthy cells, and it essentially creates kind of like a lock and key mechanism, right? So whenever the antigen has immunogens and it attaches right here, it can trigger the immune system or immune response, right? So these antigens can be consisted of proteins, carbohydrates, lipids, nucleic acids, and they're essentially found on the surface of these cells, like how I mentioned earlier, right? So this is the cell, and the antigens are at the surface, right? So they can consist of a bunch of different types of molecules. We just put carbs, proteins, and nucleic acids. So like right off the back, one of the ones that I find pretty common is bacterial. So I want to say LPS, right? LPS is a lipopolysaccharide, right? And a lipopolysaccharide is a form of fat that can be found. It is essentially an antigen that is found at the surface of certain bacteria. So, for example, this bacteria can be tuberculosis, otherwise known as TB. TB has a lipopolysaccharide found at the surface has a lipopolysaccharide, which is an antigen found at the surface of this bacteria, and it can then create an immune response to the person, leading for you to become sick, right? So that's that. More so, antigens can also be recognized by certain cells called T lymphocytes, right? So this cell right here, which is an antigen that I just drew for you guys, it is then being caught by a T lymphocyte. T lymphocyte, right? These T lymphocytes essentially play a key role in identifying antigens and present them to other cells, right? So whenever they present them to other cells, they're usually in protein fragments whenever they present them, right? So it doesn't necessarily get the whole cell. It just gets a little piece, which is the antigen, and it then presents it to its other friends so they can then be dissected and identified whether it's a threat or not. Okay. So whenever it's sent out to other cells, these antigens, like how I mentioned earlier, are protein fragments, and they can be presented to these molecules called MHC. MHC essentially stands for, so let me put an arrow. It presents it to this molecule called MHC, right? MHC essentially stands for major histocompatibility complex. Complex, right? MHC stands for major histocompatibility complex, and it's essentially a set of molecules that's made up of different types of stuff. So this MHC can be divided into two different types of classes, right? So we have MHC class 1 and then we have MHC class 2. So MHC class 1 and 2 are different from each other. MHC class 1 is primarily presents antigens from inside the cell to cytotoxic T cells, right? So it sends it out to cytotoxic T cells, right? Kind of like how you guys saw, well, this was a T lymphocyte. Cytotoxic T cells is the use of MHC class 1, and it essentially leads to the destruction of infected or abnormal cells. So it leads to destroying infected or abnormal cells. So an example of this can be something like, let me say something like HIV, right? HIV infects the cells, it leads them to become bad, and it then kills it, right? Or you have something like an abnormal cell, such as a cancer, mutated cell that has no limit for replication, and it kills it, right? So the cytotoxic lymphocyte can kill either one of these two cells that are infected or abnormal. So, and this is going to be through the use of MHC class 1. So that's that. Whereas on the other hand, we have MHC class 2, and essentially these molecules present antigens from outside the cell to helper T cells. So whenever this occurs, it also initiates a coordinated immune response against extracellular pathogens. Right, so if I draw that for you guys right now, we're going to present the pathogens. Excuse me. Let me get some water real quick because it's okay. All right. Sorry about that. So I kind of run out of breath a little bit every now and then. But so right here, let me draw this for you guys. MHC class 2 is going to be like this. So we have a dendritic cell. Dendritic cell has those right there. And this is going to be MHC class 2. Right, so this MHC class 2 already has the antigen inside of it, and it's then going to be sent over to the CD8 T cell for cross-presentation, right? So whenever the CD8 T cell picks it up, it's going to then also have a co-stimulator attached to it. All right. So whenever this happens, it's going to recognize it, and then it's going to create a T cell response so they can then send out cells to kill that. And let me draw this for you guys right here. So this CD8 T cell is virus specific. And it's a CD8 T cell. And then over here, this is a co-stimulator. All right, so that's that. So the MHC class 2 essentially has a dendritic cell that presents it to a CD8 T cell, right? So the dendritic cell sends it over to a CD8 T cell so that it can then be sent over to create a T cell response, right? And T cell response is going to help is essentially a specific type of immune response that works more efficiently, so they can then get rid of the pathogen that's inside the body. Right. So that would be that. Now, yeah. So essentially, these two types of MHC molecules are essential for the immune system. since it can distinguish self from non-self and mount appropriate responses against antigens and abnormal cells. So depending on the type of pathogen or how it's in your body will depend on the type of MHC that's going to be used, right? So it's pretty important. So next, we're going to be going into antigens recognized by T lymphocytes. So essentially, what T lymphocytes are, or otherwise known as T cells, I can draw it right here for you guys. Oops. T lymphocytes, or T cells. So T lymphocytes or T cells play a very critical role in identifying antigens and present them to other cells, kind of like how we saw here earlier, right? So the T cell response can then send the message out to other cells to create a more effective response. And these antigens are usually protein fragments and are displayed on the surface of cells through specialized molecules. These specialized molecules are MHC, like how I mentioned earlier also, right? So all in all, T lymphocytes are very important. So going into the structure and function of MHC molecules, let me draw this right here again for you guys. MHC. So MHC molecules they're kind of like presentation platforms, right? They carry antigen fragments from the surface of the cells and they have recognition. Or well, they carry the antigen fragments to the surface of the cells for the recognition of T cells, right? So it's kind of like how we saw right here through the use of the MHC right here, it was then able to present it to the T cells so they can then create an immune response and help out with the body performing more effectively and fighting off the pathogen, right? So that's that, and it's really important. So there are two different types, class 1 and class 2, like how I mentioned earlier. Class 1 would essentially be present with class 1 molecules for MHC would present antigens from inside the cell, whereas class 2 molecules would present antigens from outside the cell. So that's kind of like how we saw here. Class 1 from outside of the cell, class 2 from inside of the cell, right? Class 1, and then class 2, inside the cell, outside of the cell. And that's essentially used for the different types of MHCs. As for capturing, processing, and presentation of protein antigens, antigen presenting cells, otherwise known as APCs, they can be different types of cells. So I can draw it right here for you guys. APCs, APCs, otherwise known as antigen presenting cells, consist of dendritic cells, macrophages, and B cells. Right. So APCs consist of these three different types. And essentially what they do is they present the antigens to different types of cells so they can become recognized and initiate a proper immune response. So the way on how they operate is they engulf these pathogens and break them into smaller protein fragments. Whenever this occurs, it displays the fragments onto the surface of MHC molecules. So, let me get some water real quick. So the dendritic cell right here, I can draw it out for you. It's going to consume this pathogen. It's going to go like this, right? And it's going to have it inside of it. And it's then going to present it to uh, to its CD8 T cell. Right. So that's essentially what happens. So this process right here uh, is this one right here. So this can be either the macrophage, the B cell, or the dendritic cell, right? And it all does the same thing in sending it out to the CD8 T cell so that it can send out a T cell response. So, for antigen recognition by B cells and other lymphocytes, B cells are essentially equipped with B cell receptors, kind of like T cell receptors. So, essentially, what they do is they directly recognize intact antigens like proteins. When an antigen binds to a BCR, it activates the B cell to divide and produce antibodies specific to that antigen. Other lymphocytes, including T cells, also play a role in recognizing antigens through T cell receptors. Right. So that's a little bit of that. So we have BCRs, well, we have T cell receptors, and we have B cell receptors. And they both have antigen recognition. Right. So so that would be that. Sorry. It's getting kind of tiring trying to talk so much and keep up. So now we know that about B cells and T cells. What I can then go over now is the way on how it detects these different types of pathogens. So whenever we detect these different types of pathogens, like tuberculosis, the lipopolysaccharide, right? Essentially, what's going to happen is this pathogen right here. Sorry, this pathogen right here. I'm going to come down and draw it right here. The pathogen has what is called an epitope, right? An epitope. So this epitope is essentially what triggers the immune response. So we have the pathogen. So we have it in three steps. We have the pathogen, which is the overall virus, and then we have the antigen, right? And then at the tip of the antigen, we have what is called the epitope, right? So this epitope is essentially what's going to be causing the immune response. So if I do something like this right here, it is going to be this special figure that attaches to what is called a peritope. So this peritope is part of what is the cell's receptor. So we have the epitope and we have the peritope, or paratope. All right. So if I draw this on a different scale, we have the overall cell, and then we have the receptor. And on here we have the epitope. All right, so the cell essentially has these surface cell proteins, and they're going to attach. Well, the epitope is going to attach to the surface cell proteins, and that's how it initiates the immune response. So on the B lymphocytes, on the B cell, on the B cell, sorry, I can't talk. On the B and T lymphocytes, we have different types of receptors. So I can draw them for you as well. B lymphocyte and T lymphocyte. Right. So so this essentially helps with the recognition of antigens uh, through the use of antigen receptors and lymphocytes specifically. So for the B lymphocytes, we have a receptor like this. I'm going to try to draw it as best as I can for you guys. We have another one right here. Okay. All right, there you go. So you can kind of see right here, this is the antigen binding site. Like that. You can see how it has kind of like a little smooth shape. Antigen binding site. And we have different parts of this chain. So so yeah. So on the antigen binding site, which is at the top, we have that part, and then we also have several other parts as well. So we have two different types of regions. We have what is called the variable region, and then we have the constant region. Constant region. So variable region is going to be the region on top. Constant region is going to be the region on bottom. Top and bottom, right? So we also have what is called the heavy chain. The heavy chain is going to be this part, and it is essentially the core, right? So heavy chain, right? And then we also have what is called the light chain. Right, so depending on the different type of receptor will determine on how the on how, what depending on the different type of receptor will determine on how it is laid out, on how it is created. So sometimes the antigen binding site, instead of a perfect U, let's say for example, it could be kind of be like a W, or it could be a square, or it could be a V. You know, sometimes there are different types of variable regions depending on the type of receptor, and this can also dictate what kind of pathogen would be able to attach to that specific cell, right? Because I mean, sometimes if you think about it, we have different types of viruses such as the flu. Okay, the flu, which would only attach to epithelial cell within the respiratory tract, right? It's a respiratory virus, and because of that, it would only attach to epithelial cells within the lungs. Okay, so if I do some lungs right here, the esophagus, okay, that's the esophagus on top, and on bottom is the lungs. So essentially, what happens is these antigens attach to epithelial cells within the respiratory tract that have a certain binding, or that have a certain antigen binding site, and this allows for you to only get sick mainly within this region. So same thing with strep throat, right? It attacks the epithelial cells within your respiratory tract. That's why you get a sore throat and those cells become damaged and infected. And that's also why you don't have like on why you get a rash on your hand whenever you get streptococcus, because of course it doesn't affect the epithelial tissue cells on your hands, on your skin. It's only the ones in your respiratory tract. So that's a little bit of insight about that. Moving on to the T lymphocytes, it's also the T lymphocyte. We're going to do a T cell receptor. Oh, also I want to mention this is called a surface immunoglobulin. Surface immunoglobulin. All right. And then this one over here is a T cell receptor. So that's that. Now, in order to draw the T cell receptor for you guys, it's a little bit more. It's the shape of it is a little bit different. This is also because it differs in the types of antigens they recognize. Kind of like how I mentioned to you earlier, depending on the shape will determine on different kind of antigen it recognizes. Same thing over here. So, so this one is still going to have a constant region and variable region as well as an antigen binding site, but it will be drawn kind of like this. I am not the best artist. If you have seen from my previous videos, they are not the best, but still trying. Okay, so like how I mentioned earlier, we have the variable region. Var oops. Variable region. And then we have the constant region. Okay. And then we also have the antigen binding site. And then of course right here, well, let me draw this a little bit. Oops. So on this right here, we have what is called the transmembrane region. So this is essentially what we can think of as the cell wall, right? Trans man transmembrane region. Region, right? So um, that's pretty much what a T cell receptor is and how it operates. Now, I can also I can also draw you a little bit more of an in-depth picture as well. So sorry if I'm not talking that much. My throat's drying out a lot also from talking. Okay, so here we have a T cell receptor. TCR. And then we have MHC. Remember, MHC is the one for recognizing. So the MHC essentially has, it's kind of like the key and lock mechanism as well. Let me draw this in a different color. All right. So that's that, and then we also have the anchor residue or peptide, which is essentially what binds the MHC to the T cell receptor. Right, and then depending on the anchor will depend on how efficiently it's going to be attached to it. All right, so that's that. So we have this is essentially right here. This is the T cell contact residue of peptide. So, man, T cell contact residue of peptide. All right, and then over here we have kind of the same thing except it's a polymorphic residue of peptide, PRP, just to make it short. And then over here is also the same thing. Anchor residue of peptide, right? All the same thing. They're just different ways of attaching the T cell receptor as well as the MHC together so they can properly attach to it. So, in addition to that, for the MHC, we also have two different types of receptors, or two different types of antigen presenting cells. Well, we have antigen presenting cells for two different types of MHC classes. So, as I mentioned up above, we have MHC class 1 and class 2, depending if it's internal or external. And then, let me write it right here. A, P, C, antigen presenting cell. So for these APCs, we have MHC class 1 and MHC class 2. Right. So down below, I can draw the transmembrane for you guys. And then we are going to have different types of MHC. All right, so we got this one, goes like this. That is very bad. I am a terrible, terrible, terrible drawer. So that is one. And then MHC2 is kind of the same thing, right? Except it's just a bit more different. Instead of having the circle at the bottom right, it has squares. And this essentially determines what kind of antigen it can receive or detect. Right? So you can see how it has two squares and this one has a square and a circle. That is pretty much going to determine what kind of antigen it can detect. So those are two different types of MHC classes and how they're drawn out. So on top of that, we also have the function and display of microbial antigens. So essentially what happens is in different parts of your body, we have infections that can occur. So for example, we have the infection of, let's say for example, someone has strep throat, right? So that's in the respiratory tract, well, primarily in the esophagus, right? Okay, so we have a bunch of germs. Right, so we have those. And essentially what's happening is the microbes are going through the epithelial barrier. Whenever they go through the epithelial barrier, we have macrophages that are present on site, right? So let me draw a microbe. And then here we have tight skins or tight junctions to create that physical barrier for immunity, innate immunity, right? So we have those macrophages that can then consume. They can then consume the pathogen, right? And because the macrophage is an antigen presenting cell, um, sorry, well, it could be a macrophage, a dendritic cell, or a B cell. Either way, it's gonna present this all the way to the lymphatic vessel, right? So the macrophage, send it to the lymphatic vessel. Kind of looks like a little bean or something, and it sends it through. It sends it to the lymphatic vessel through the lymph nodes. So this APC has the pathogen with it, to where it will then be processed within the lymph node and then absorbed And essentially what happens when it absorbs this pathogen, the lymph node will collect these antigens from the tissue, and it will create an immunologic memory so they can create antibodies. We'll talk about this soon, if not in the next chapter for sure. So let me write it for you right here. Lymph node collects antigen from tissue. All right. So that's how that goes. Um, Additionally to that, we also have cell-free antigens that are created whenever the macrophage sends it out to the lymphatic vessel. So like this right here, cell-free antigens. These essentially what happens is these antigens can then be sent out into the bloodstream, right? Whenever these cell-free antigens get sent into the bloodstream, they are then presented to the spleen, right? So let me draw out the spleen real quick. That way I can tell you guys what it's all about, how it functions, the purpose of it. So it then sends out circulation to the spleen, and essentially what happens is bloodborne pathogens are captured by these APCs in the spleen, to which it can then eliminate them, right? So the spleen is also plays a very important role within the immune system, and it's critical for filtrating and, and killing different types of pathogens. So as I mentioned earlier, we have the B cells, we have the T cells, right? And the B cells are created in the bone marrow, and the T cells are created in the thymus, and then on top of that, we also have these are the primary locations where they're created, and they can then get sent out to secondary locations, which are known as peripherals, which is stuff such as the lymphatic vessel. We just put lymph node as well as the spleen, right? So that's part of it, and yeah. So more so, a little bit about dendritic cells is whenever they're immature, they tend to reside within the epithelia of the skin. All right, so I do this, I can put a dendritic cell. All right, so they reside within the epithelia of the tinge of this. Sorry, I cannot talk right now. They reside within the epithelia of the skin tissue, right? This is whenever they're immature. However, whenever they mature, they then get sent out to grow within the lymph nodes, right? So this is immature dendritic cell, and then it gets whenever it's matured, it then gets sent over to lymph nodes so they can then continue to keep on growing, right? So yeah, that's that. Um, let's see, what else can I talk about? So I can talk about the cross presentation of antigens to CD8 T cells. So let me draw out the chart for you. So we're gonna have a macrophage, right? This macrophage is already gonna have the antigen, the viral antigen inside of it, and it's gonna present it to the dendritic cell. So let me have the antigen, and these are the receptors. And this is going to be, since it's already inside, since the antigen is already inside of the cell, it's going to be sent over to the dendritic cell through class 1 MHC. Class 1 MHC, right? And essentially, fragments of these cells are going to be picked up by host antigen presenting cells. So these host antigen presenting cells can also be known as dendritic cells, right? Dendritic cells. All right, these dendritic cells then pick up the viral antigen to where they can present it to a CD8 T cell. Whenever the dendritic sends it over to the CD8 T cell, this would then be classified as MHC class 2. Class 2 MHC, right? So the viral antigen will then enter cytosol, and it will then be sent over to the CD8 T cell, where it will then be attached to it through the use of a co-stimulator. Okay, and this is the co-stimulator right here. All right, so whenever that happens, it's going to then activate a T cell response. CD8 T cell. Right. So that's pretty much what happens, and yeah. So that pretty much concludes the video presentation that I have for chapter 3 on antigen capture and presentation to lymphocytes. And I want to thank you guys for watching.